Previously on Science.
We discussed the process of a germ cell that would undergo mitosis and meiosis.
Due to popular requests, we have a special episode for you today on DNA.
Today's topic is the DNA replication process.
My name's Hannah.
My name's Shahana,
and we'll be your hosts for today's segment. DNA replication occurs during the S phase of interphase, which was covered in the last month's segment.
When learning about replication, it's important to note the significant roles of speed and accuracy in the process, which is facilitated by several specialized proteins that we'll get into later.
The process is initiated at a specific nucleotide sequence known as the replication origin. In eukaryotes, there are many replication origins, while prokaryotes only have a single replication origin. Another thing to note is that DNA replication is semi-conservative, meaning that one half of the original parent is used and the other half is new DNA, resulting in the creation of two daughter molecules.
Keeping in mind the double-stranded structure of DNA, the enzyme helicase, represented by Pac-Man, essentially unzips the DNA in half, creating a Y-shaped fork from the double helix. The unwound area is called the replication bubble.
This enables elongation to occur starting with RNA primase, which is represented by Q*bert. RNA primase serves as the starting point for the attachment of nucleotides, which DNA polymerase will work from.
DNA polymerase catalyzes elongation in the 5' to 3' direction. It is inserted into the replication bubble and continuously adds nucleotides to the leading strand, creating a strand of DNA. As it approaches the helix, DNA is then replicated on the lagging strand.
Replication on the lagging strand is different. Unlike the leading strand, the lagging strand is replicated in opposite direction that the fork is proceeding through. Still in a 5' to 3' direction though. The lagging strand is tricky because it can only go until it hits a certain point that has already been replicated, creating segments known as Okazaki fragments.
Every time DNA polymerase jumps from segment to segment, new RNA primer is needed for each fragment. Once the DNA polymerase has done its job, ligase, which is represented by the glue monkey, can now begin its job. Ligase acts like a glue and glues the parent and new DNA together on the lagging strand.
This, however, does not mark the end of DNA polymerase role. So it acts as a proofreader that recognizes whether or not hydrogen bonding is taking place between the new base and its complement on the other's original strand. When a mismatch is found, the absence of hydrogen bonding, then the DNA replaces the incorrect base from the new strand and adds a correct base, using the parent strand as a template.
Once DNA replication is complete, the cell can now move on from the S phase to the G2 phase and then continue its journey through mitosis.
My name's Hannah.
And I'm Shahana.
This concludes this month's special on DNA replication.